Alright, hello everyone. This is the first video that we have. I hope this is a helpful tool to prepare for our tests, our exams, and if you miss class, it's a way for you to learn at home or to review on your own. So today we're going to be talking about tonicity and about endo and exocytosis. Okay, so everyone should have notes on this. The slides are on Google Classroom, and all the information from the slides is in your printout. So let's get started. So tonicity is the measurement of solute in water. So how much concentration of something do we have in water? Okay, the solvent is what the solution or the solute dissolves into. For example, if we are going to make salt water, water is the solvent. Okay, water would be the solvent, and salt would be the solute. Okay, all right. There are three types of tonicity that a solution can have to another. Okay, so we have isotonic, hypertonic, and hypotonic. We're going to talk about these three. Again, this is a review. The main point here to focus is that tonicity is measuring the concentration, so how much of the solute we have in a solution, not how much water there is in the solution or the solvent. Okay? So the three types that we had, like I said, is isotonic, hypertonic, and hypotonic. And we're gonna skip through this slide because I think it's easier if we look at the examples. Okay. The main thing to know here is that animal and plant cells, they change shape and size due to osmosis. And if you remember, osmosis is the movement in and out of water within a cell. And osmosis is affected by the kind of solution that a cell is in, if it's in a hypertonic, hypotonic, or isotonic solution. So when a cell is placed in an isotonic solution, it stays the same. Isotonic means that the concentration inside the cell and outside the cell of the solutes are the same. So water doesn't go in or out, it stays the same. It doesn't shrink, it doesn't grow, it stays the same size. Now, when a cell is placed in a hypertonic solution, there's a higher concentration of solute, of the thing that you're mixing into your water, on the outside of the cell. Okay? So there, for example, in this example, if we have salt here and salt inside the cell, in this solution of salt water, there's more salt water outside. So what's going to happen is that water will leave the cell in a hypertonic solution, okay? And the animal cell will shrink, it'll get smaller. And with plant cells, the cell wall stays the same. But the cell membrane, this part right here, the membrane that's around a cell, it's on the inside of a plant cell, it pulls away from the cell wall and the cell wall shrinks. So the main thing you have to know is in a hypertonic solution, there is a higher concentration of solutes on the outside. So water is going to leave the cell. Water is going to leave the cell. Now in a hypotonic solution, it's the opposite. Okay? There is a higher concentration of solute inside the cell than on the outside. So in our example, there is a higher concentration of salt inside the cell than there is outside. So water is going to go inside the cell, and it'll it'll make the cell kind of like fill up, similar to a balloon. And in some cases, the cell can actually pop. It's very dangerous and painful. The same thing will happen with plant cells, but they won't pop because of the wall, the cell wall. The cells will actually they'll get full, but they'll hold the pressure. Okay. This is called right here turgor pressure, and that's actually what helps plants stand up once their cells are full of water. Okay, in this example, we saw that there are 3 solutions here, and these are cells. These are human cells. So here we have cells that are in over here, hypertonic. As you can see, the cells start to shrink. This is an isotonic solution. Nothing changed. They're happy cells. They're good. They're healthy. Everything's great. And over here, you have cells in a hypotonic solution. As you can see, these red blood cells are really full. They kind of look like little balloons. And that's the main point that you have to remember here. In a hypertonic solution, water is going to leave and it's going to make the cells shrink. An isotonic solution, there will be no change. And then in a hypotonic solution, right over here, the cells will fill up with water. Now, this is really important when you're getting an IV. Okay, it's very important that the solution that you're putting inside is isotonic to your blood. If not, the blood cells can shrivel or they can swell. So the same thing. So if your blood, if you're putting in medicine through an IV that is isotonic, nothing will happen. You'll take the medicine and it'll be great. If it's hypertonic, your blood cells could shrink like this. And if it's hypotonic, they can get like this. They can swell up and it can be very painful.